Okay, so now that we have our DNA written in, again by triplet, 1, 2, 3, 1, 2, 3, and our RNA figured out, we have to find out what the amino acid is. Okay, now I'm going to attach this sheet, oops, got two papers there, this sheet to the Google Classroom. This tells you all of this business about transcription and translation and that sort of business. What I need you to do is understand that we're going to be using the Universal Genetic Code Chart. I'm going to try and zoom in here a little bit. I'm working within the limits of my camera here. Now, when we converted those way back here, our first RNA sequence was GGC. It's like Little League. There's always that one kid that hits the ball and runs backward on the bases. Well, that's kind of what we're doing here. We're going to start at first base, then to second base, then to third base. So GGC, we're going to start first base. There's our G. Second base, here's our G. Come together and you'll find GGC, which gives us gly or glycine. So back to this, GLY. That's an actual amino acid. If you look on the back of maybe some vitamins or like weight gainer or some of that powerlifting stuff, you might find that. Our next sequence is AUC. See if you can do it. A, U, bring them together. C gives us ILE. Now, ILE is isoleucine. It's another chemical, so we have ILE. Our third is GUA. GUA gives us valine, VAL. So these are just abbreviations for the amino acids. Next is GAA. It's not Girls Athletic Association. GAA gives us glutamine, GLU. And one more, UGU. Back to this. Okay, UGU gives us cis or cysteine. Okay, CYS. Now, this means nothing to you right now, but here's what we're going to do. We're going to go back to those pieces that we already cut out, and I'm going to find a GLY. Now, it's best to have them all laid out. I have a GLY right here. Now, I'm going to take away this paper, the cow insulin, for now. Okay. So GLY, here's Mr. GLY. I'm going to move this, let me see if I can zoom out. Might make it a little bit easier. Might be tougher to read, but I'll kind of walk you through it. GLY, there's our first amino acid in the cow insulin gene. Next is ILE. So I'll go over to my pile, which is over there, and I'll find my ILE. So these have to fit together. Here's what you do. I'm just using a little bit of tape. You can use a glue stick, that's fine. On your ILE, make it so that you're continuing from that arrow in GLY. It's going to be stuck to the table now. Okay. Now, our next is valine, VAL. Here's a VAL. Give another little piece of tape. VAL. You have to continue this line, remember. Okay. Arrowhead to arrowhead. So you're always going to be taping the next amino acid onto that X. Okay. Next is GLU. Now I will tell you, on this sheet, the GLY, the GLN, and the GLU look very similar. So you have to be careful when you do it that you get the GLU, which is what I'm looking for here. Okay. They're not similar in shape, they're similar in look, okay, as far as the actual writing. So there's our GLU, and that's going to continue the line once again. So see what we got. We have this kind of folding, twisting shape starting to form. And our last one that we did was CYS. And CYS will start us in another direction. So it's basically going to form this long, twisted, for lack of better terms, snake of paper. And as long as you're continuing this line, okay, as long as you're continuing this line right here, here's where we started, you'll see that the arrow always goes, and the next one would be taped on here, whatever it might be, okay? That's what you want it to look like. Now, once you get this entire thing built, all 21 sequences, or triplets, or codons, whatever you want to call them, you're going to have something that you should be able to get rid of our amino acids. Make sure my camera doesn't fall. Since we're doing the cow insulin, I'm going to pick all this up, hopefully not tearing it apart. Here we go. So let's pretend the entire thing is built. You're going to somehow position it on your paper, and it should all fit. In the past, it's all fit on the paper. If you cut them out carefully, this is what you're going to have, okay? It's up to you. Glue or tape, doesn't make a difference to me. As far as turning in the lab, keep the paper copy. We might be able to use your Chromebook to take a picture of it and include it, that sort of thing. It's the same procedure for the human insulin, same exact procedure. Okay. The other thing before I go that I want to show you is, I showed you this universal genetic code chart, the square one, okay? There is another, and it's actually, I prefer this one because it's more like playing darts, okay? It's a universal genetic code chart where you start in the center and then you move out. For instance, on ours, where's our sequences here? Sequences. Our first sequence was GGC. Okay, so you would go to G in the center, start in the center. Here, let me get the point of the pen and be easier. G, G, C gives you glycine. Huh. Imagine that. AUC, A, U, C gives us isoleucine, ILE. Look at that, okay? So you can use either, and if you just Google Universal Genetic Code Chart, you will find that. If you can't find it, you're not Googling hard enough. I will try to splice these two videos together, but it'll probably be two separate videos, and you'll have to watch them both. But that is how you do the cow and human insulin comparison lab. The same thing holds true for the protein synthesis and words lab. It's the same process, except instead of finding little pieces of paper, you're making a sentence. All right? Take care, everybody. Give it a shot. That's the best I can ask. And I'll see you soon. Bye-bye.